The interpretation and scoring of the tissue samples depicted in this video are for illustration purposes only and do not replace a physician's medical training, judgment, and experience in this matter. Human epidermal growth factor receptor 2, or HER2, is a protein found on the surface of a cell. Many cells contain HER2 protein, but at normal levels. The number of HER2 genes in a cell usually determines how much HER2 protein is expressed on the cell. Too much HER2 protein causes cancer cells to grow and divide more quickly than normal cells. HER2-positive breast cancer is considered aggressive because it grows and spreads quickly. As you can see, different regions of the tumor may express HER2 at different levels. This is the concept of tumor heterogeneity that may impact accurate HER2 testing. Initial testing for cancer can be done using a core needle biopsy or a resection. ASCO-CAP recommends that resection samples be preferentially used for HER2 testing rather than core needle biopsies because of the likelihood of artifacts which might confound interpretation. Pathologists should use discretion in choosing the best sample for testing in each patient. However, this is only a small part of the HER2 testing process. We will move forward as if a resection was the extraction method used. Now that the sample has been removed, it should be fixed within 1 hour. The pathologist will place the sample in formalin, which will keep the specimen from degrading and becoming unusable. Before it's tested, the sample should be fixed for no less than 6 hours and no more than 48. This allows for the fixation process to occur. The sample should be embedded in paraffin and then sliced for staining. Ideally, ASCO-CAP guidelines involving sample handling were followed. At this point, the pathologist has used the recommended type of formalin to fix the sample, placed the tissue in formalin within an hour of extraction, allowed enough time for the formalin to penetrate into the sample, embedded the specimen in paraffin blocks, and sliced the tumor samples for staining. Now that the sample has been correctly prepared, it is ready to be tested for HER2. The most commonly used FDA-approved methods for determining HER2 status are IHC and FISH. IHC testing shows the amount of HER2 protein on the surface of a tumor cell, as well as the distribution of highly stained cells within the sample. IHC stains the sample using HER2 antibodies and a chemical detection method. IHC test results are scored negative 0 to 1+, equivocal 2+, or positive 3+. An IHC score of 3+ means there is excessive HER2 protein expression. Specimens that are scored IHC 2+ are considered equivocal and, according to the ASCO-CAP guidelines, should be retested using another FDA-approved test. Another FDA-approved test is FISH. FISH measures the number of copies of the HER2 gene in the nucleus. The pathologist will count the number of genes in the nucleus of a set number of cells to determine the HER2 status. FISH tests are scored as a negative ratio, equivocal, or positive. IHC and FISH are both appropriate testing methods, but when the first test is equivocal, a second test using another method should be used. If tissue handling isn't carried out correctly, the results of the HER2 test may be inaccurate. These are some of the potential problems that can arise for both IHC and FISH testing. If the tissue isn't placed in formalin within 1 hour, the sample may degrade, leading to a weak signal, and the sample potentially becomes unusable. If the sample is left in the formalin too long, it can potentially create a false negative result because the protein and DNA may be negatively affected. formalin doesn't make it all the way through the sample, it may not be completely fixed, which can lead to improper staining. Core needle biopsies may cause artifacts that can be seen under the microscope. They can be falsely interpreted and create a false positive result. Any sample with artifacts should be rejected. Tumor heterogeneity is an important issue in testing for HER2. Tumor heterogeneity is when the cells within the tumor may not all be the same, which can lead to one part of the tumor to test as HER2 positive, while another part will be HER2 negative. This can lead to a false negative result. Due to tumor heterogeneity, all of the pre-analytical steps could have been done properly, but mischaracterizations may still occur. There are ways to avoid this pitfall that will be discussed later. Discordance is when two HER2 tests don't match. Two potential reasons could be that either one test was flawed or the results are due to tumor heterogeneity. Limitations in assay precision make it inadvisable to rely on a single method to rule out HER2 positivity. Only one positive test is needed to indicate HER2 positive status. 200,000 patients are diagnosed with breast cancer every year. Approximately 25% of breast cancer patients are HER2 positive. Treatment decisions rely on accurate tests. Following testing guidelines may help ensure more accurate test results. Following fixation guidelines recommended by ASCO-CAP is a step in the right direction towards accurate HER2 testing. Excision samples, when available, are preferred because a core needle biopsy may not represent the whole tumor, and tumor heterogeneity could potentially create false negative results. Minimize false negative and false positive results by using the appropriate HER2 tests. The challenge of accurate HER2 testing may be overcome by using proper sampling and testing from multiple tumor blocks. IHC and FISH are complementary tests. When a first result is equivocal, the other test should be used. The interpretation and scoring of the tissue samples depicted in this video are for illustration purposes only and do not replace a physician's medical training, judgment, and experience in this matter.